Hello everyone, I am Dr. Sonali and this is the part 2 of my lecture on vaccines. The link for part 1 is given here. Now in today's lecture, we will be talking about cold chain, vaccine vial monitor, newer vaccine technology and national immunization schedule. Also by the end of today's lecture, I'll also tell you how to write short notes. Okay, that is how to approach short notes. Okay, let's start. Now we all love eating ice cream, right? Everyone loves ice cream. Now, what happens when the temperature of this ice cream is not maintained? It gets ruined. So do our vaccines. That's why to maintain the temperature of the vaccine, what do we require? We require cold chain. Okay. So cold chain refers to a system of transport, storage and handling of vaccines starting at the manufacturer level and ending with the site of administration of the vaccine to the client. That means it is like a chill chain, okay, a chill chain starting at the manufacturer level, which is also present at the storage and the PHC till clinics, and it ends at the patient's arm or wherever is the site of that vaccination. Clear? Now, what are the ideal storage temperatures for your vaccines? We'll see it. The first gang. The first is your frozen vaccine gang, which is minus 15 degree to minus 20 degrees Celsius. Now, the two examples of this frozen vaccine gangs are OPV and measles. Okay. Now, both are extremely heat sensitive and should be stored at minus 15 to minus 20 degrees Celsius. But among these two, OPV is the most sensitive. Okay, it's the most sensitive to heat. Clear? Now, the second gang we have is the refrigerated but never frozen gang. Such vaccines can be refrigerated, that is at plus 2 to plus 8 degrees Celsius, but should be never frozen. Why? Because freezing will damage the protein. Okay? It damages the protein and causes vaccine failure. Now, whenever you are handling this box, okay, so what do you say it to anyone handling this box? You say you have to give something as a caution. So you say, be careful that this health box doesn't freeze. Okay, this is like a warning which you have to give. Now let's see those vaccines which should be refrigerated but never frozen. That is BCG, combined DPT, it is tetanus toxoid, this is adult diphtheria toxoid, this is hepatitis B, this is Haemophilus influenza B, and D stands for diluents. Okay. Now, let's see the third gang. That is the light sensitive one. The ones which are like vampires, which are extremely sensitive to light. The example, BCG. So, BCG should always be stored because of this in amber colored bottle or should be covered by a dark paper. Okay. So, this was everything about your cold chain. Now, how do you know that your cold chain is being maintained or not? How do you know that all of this is functioning? For that, we have vaccine vial monitor. It is a tool to monitor the stability or potency of a vaccine and to check the efficiency of the cold chain. Think of it like a heat sensitive sticker. Okay, it's a heat sensitive sticker. And what all do you know from this sticker? The first point is that whether it has been affected by heat or not. Second, whether the cold chain was maintained or not. Third part, is the vaccine stable or potent? Okay. Now these are the things which you can know by vaccine vial monitor. What you cannot know, okay? What it doesn't tell us. It doesn't tell us about the expiry date of the vaccine. You have to check it by yourself. It doesn't tell us about whether the vaccine is contaminated or not. Okay. Now, what does a vaccine vial monitor looks like? Okay. It looks like this. Can you see this? This heat sensitive sticker which is on this vial is the vaccine vial monitor. Now, let's see the parts of this. It has an outer circle which is blue in color. Okay. And an inner square which is white in color. Now, this inner white, as it is exposed to heat, it changes its color. It becomes darker. Okay. So, for a vaccine to be stable or potent, your outer blue should always be darker than your inner square. Clear? So, WHO has come up with certain stages of your vaccine vial monitor to tell us till what stage it will be working. So, we'll see your stage 1, the outer circle is blue, the inner square is white. Here you can use the vaccine, it's stable. Second stage, your outer circle is blue and inner square has become light blue. It's still working. Okay, good. Third stage, the outer circle is blue but the inner has become a bit darker than the previous one. It has almost become equal. You cannot use. Now, the last part, that is the fourth part, the outer circle is blue and the inner circle has become darker. Absolutely not. Okay. So, this was all about your cold chain and vaccine vial monitor. Now, let's see some newer vaccine technology. Okay. The mRNA vaccines. Now, before we know what are mRNA vaccines, let's see what is mRNA. Now, mRNA or messenger RNA is a molecule that contains the instruction or recipe that directs cells to make a protein using its natural machinery. So, if your cell is a cook, okay, and it has to make a protein dish, then this mRNA is like a recipe, okay, it's like a recipe which directs the cell to make this protein dish. So, sorry for the spelling, this protein dish using only the natural machinery, that is the natural ingredients which is present in its kitchen. Okay. Now, let's see what is this mRNA vaccine then. Your mRNA vaccine or messenger RNA vaccine are a breakthrough. It is quite new. Okay. It was a breakthrough vaccine that instructs cells in a body to produce a harmless piece of virus. That means what it is doing? It is giving the recipe or like a code, a cheat code to your body to produce a harmless piece of virus. Like for your COVID-19, the spike protein found on the surface. This spike protein found on the surface. Okay. So, the mRNA are breakthrough type of vaccine that instructs cells in the body to produce this harmless piece of virus like spike protein found on the surface of your COVID-19 virus. This small piece of virus triggers our immune system to recognize it and produce antibodies in response. Clear? This is the definition of your mRNA vaccine. Now, let's see what happens after you are vaccinated with mRNA vaccine. The mRNA enters your cell. Now, this mRNA is usually in a lipid nanoparticle, that is capsule. It is enclosed in a capsule. Why? Because you see, if you just give the naked mRNA, it's very fragile and it will be destroyed by our natural enzymes. Okay, that's why it's enclosed in a lipid nanoparticle. Now, what will happen? Our cells will read the mRNA and make a harmless viral protein like the spike protein of SARS-CoV-2. Okay, it's harmless. It doesn't cause any harm to our body, but it will do what? It will train our immune system, train our army. Okay. Next, your immune system sees this foreign protein and goes intruder alert. It goes into an intruder alert mode and starts the training of your army. Now, if the real virus attacks later, your body already has a trained army. Okay? This was what happens after the vaccination. Now, let's see why is it so revolutionary. Here we talk about the advantages and the application of mRNA vaccine. So, what is the first advantage? The first advantage is super fast to develop. See, you don't need to grow the virus. Remember, you don't need to grow the whole virus via cell culture or your egg culture. Okay? All you need, what do you need here then? You need is the genetic code. Okay? All you need is the genetic code here. And then once you have the genetic code and your mRNA is designed, you can have a mass production of your vaccine. Okay, that's why it's super fast to develop. Second, it's quite adaptable. See, even if the virus mutates, what you can do? Even if the virus mutates, you can change the mRNA, change the mRNA code to target the new variant. Okay? That's why it's extremely adaptable. Next, it has fewer side effects for people who are immunocompromised. Obviously, it doesn't contain any live virus. There is no live virus. Okay? So, it's very safe. You know why? Because as we have seen before, it is producing harmless protein in our body. Those harmless protein are only training our immune system. Okay? So, it has fewer side effects for people who are immunocompromised. And it has a high efficacy in preventing severe illness. Now, many clinical trials have seen that it has extremely high efficacy for preventing severe illness, more than 90%. Okay? Now, the example, the first mRNA vaccine to be approved was your Pfizer vaccine for COVID-19. Okay? Now, this was everything about your mRNA vaccines. Now, let's see a diagrammatic representation for a revision and better understanding. So, this is the viral genome. You took the mRNA part, the gene for specific protein. It is packed in a capsule, that is the lipid nanoparticle. Now, this nanoparticle is delivered by the syringe. This nanoparticle or the capsule. See, this capsule, it fuses with the cell membrane, releasing the genetic code. The code is released here. Now, instructions are read and cell makes the target protein and the vaccines are broken down after reading. So, what happens here? Cell present targets protein on surface where it is found by the immune system. After that, the immune helper cells trigger an immune response. Body creates army of B cell. Antibodies are formed, infected cells are recognized and destroyed, and memory cells are also created in case if the body encounters the virus later. Okay? Now, let's move ahead. Edible vaccines. What are edible vaccines? As you can see in this definition, they are plants. What kind of plants? Genetically modified plants. Such plants are called transgenic. Okay? Now, what do these plants have? They are genetically modified and they produce antigen. Antigens are proteins from the pathogens in their edible part. Okay? That's why the name edible vaccine. Clear? So, this is the definition of your edible vaccine. Now, imagine this potato, okay, or your banana. Not just as fruit, but your vaccine delivery system. Okay, your vaccine delivery system. What happens when you eat them? Your immune system will recognize these antigen and will be trained to, if in case in future, if it encounters the antigen again, it will attack it. Clear? This is your edible vaccine. Now, how are they made? Scientists isolate the gene that encode the antigen from a pathogen. Example, the antigen from your pathogen like cholera or your Norwalk virus. Then what? This gene is inserted into a bacterium which is plant friendly, like your agrobacterium. Okay. Now this bacterium is used to infect a plant, be it your banana, potato or tomato. The plant becomes transgenic, genetically modified, and start producing the antigen in its edible part. Clear? Eating the raw or processed plant gives us oral immunization. Clear? Now, let's see the advantages. Low cost, no syringes, cold chains or trained health workers. Second, mass production, can grow like any crop. Third, oral route, no needles, it's no pain plus patient friendly. Fourth, local mucosal immunity, especially for gut pathogen, is very useful. Okay. Fifth, it's heat stable. It doesn't need any refrigeration. Now, let's see the examples. Transgenic potatoes and tomatoes, they are used against your diarrhea, E. coli and cholera. Your edible banana against Norwalk virus, which is a cause of gastroenteritis. Clear? Now, this was your newer vaccine technology. Let's see national immunization schedule. So, I have divided your national immunization schedule according to the group of people who are getting it. So, we'll start with the first group for pregnant women. Okay, let's see. There is TT or TD first dose early in pregnancy. That is your adult tetanus can be combined with your adult diphtheria toxoid. First dose early in pregnancy. Then second dose of adult tetanus toxoid will combine with either adult diphtheria toxoid 4 weeks after the first dose. Okay. Boosters, if 2 doses are taken in the last 3 years. It is 0.5 ml intramuscular in your apana. This is for your pregnant women. Clear? Now, for infants, at birth we'll start. So, for infants at birth, what do we give? First, we give BCG, intradermal. Then we give hepatitis B, intramuscular, and OPV, that is the 0 dose OPVs, oral polio vaccine. Okay. Now, if you have trouble in remembering this at birth vaccine, I have come up with a mnemonic. Okay. See, kids are quite optimistic, right? And especially when they are born, they are quite optimistic about life. So, what we can say that this baby, ye baby hai optimistic, right? So, B for BCG, H for hepatitis B and O for oral polio vaccine. Now, let's see the 6, 10 and 14 weeks. First is the pentavalent vaccine, first, second, third dose at your 6, 10 and 14 weeks. Pentavalent vaccine contains DPT, hepatitis B and Haemophilus influenza B vaccine. Then is your rotavirus oral dose, orally. Now, your rotavirus is only given in selected states, not throughout India. Okay, only in selected states. Then you have your oral polio vaccine, first, second and third dose. Now we have these two, that is pneumococcal conjugate vaccine and your injectable polio vaccine. Now these two are given only at 6 and 14 weeks. Okay. Your pneumococcal conjugate vaccine is also given in selected states. Clear? And your injectable polio vaccine is given at 6 and 14 weeks intradermal. You know, one important point about your IPV is that previously it used to be given at 14th week. At 14th week with oral polio vaccine, third dose of oral polio vaccine, and you have to give the full dose of IPV. Okay, but this was earlier. But now what do we do is we have divided it to fractional dose given at 6 and 14 weeks intradermally. Now, why was this done? First reason, but it was seen that this latest schedule which was being followed, it provided better immunity, okay, or we can call better seroconversion. Seroconversion means that after vaccination, there is a good amount of detectable antibody which is present in a patient's blood, okay, that is known as seroconversion. That means better immunity has been developed after this vaccination. 2 fractional dose, 6 and 14 weeks given intradermally. Second, this present regimen was cost effective. It was more cost effective. Okay, why? See, if you are giving full dose was around 0.5 dose. So it is a lot, but if you are dividing it in 0.1, okay, so what will happen? You are dividing it. You are reducing the amount of vaccines to be produced. You are reducing the amount of vaccines. So it is more cost effective and cost friendly. Okay. Now, again, if you have problem in remembering it, I have again a mnemonic for this 6, 10 and 14 weeks. That is, as the baby progresses from birth to the 6, 10 and 14 weeks, it becomes pro. It becomes pro in what? It becomes pro in playing. So, P stands for your pentavalent vaccine, R for rotavirus and O for OPV. So, this pro is given at 6, 10 and 14 weeks. But this I for your IPV and P is PCV. This in playing is only given at 6th and 14th week. Clear? Now we'll move ahead. For older children, your 9 months. Your 9 months, you'll start with your measles or measles rubella plus Japanese encephalitis first dose and vitamin A first dose. Now I have a mnemonic for this also. Whenever you watch your MTV, what you can see? You can see your VJ hosting a show. M stands for measles rubella, V stands for vitamin A and JE stands for Japanese encephalitis. Now the problematic things to remember is only till 9 months. After that, it's quite easy. See, you can combine these two, 16 to 24. You can combine this also with them. So, most of the booster doses here are from the first doses of 9 months, like your measles and rubella second, your Japanese encephalitis 2 and vitamin A second dose, all from the 9 month which had the first dose. We are giving the booster dose at the 16 to 24 months, right? Also, one thing about your Japanese encephalitis, it is also given in selected state. Okay. Another just additional 2 vaccines are here, that is DPT booster 1 and OPV booster. For vitamin A, just remember this, that the 2nd to 9 dose is given every 6 months till age 5. Now, for your 5 to 6 years, it's DPT booster 2 and 10 to 16 years is tetanus toxoid with adult diphtheria toxoid. Okay. Now, let's see some clinical scenarios, which will help us understand national immunization schedule better. So, if you have a child more than 1 year of age and goes to a doctor and hasn't received any vaccine, now what to do? Answer is, you will give 3 doses of DPT at an interval of 4 weeks, measles first dose, Japanese encephalitis first dose in whichever states it is applicable. It is up to 2 years age as a catch-up immunization. This is known as catch-up immunization. Okay. Now, why only these vaccines we are choosing? Okay. Remember, your catch-up immunization is for public health prioritization. Okay. You have to prioritize public health here. You don't just have to finish your textbook schedules. So we'll see first DPT. See, your diphtheria and your pertussis both causes disease which have lots of complication. Okay, and it can also lead to death in young children. Okay, and your tetanus, there is no natural immunity against tetanus. Okay, this is not present. So that's why DPT becomes a high priority. right? Second vaccine, measles. Now, your measles is the cause of high morbidity, okay? High morbidity and mortality in children less than 5 years. So, this also becomes a priority. Third, Japanese encephalitis. Now, in the states, selected states which are endemic, okay? Now, 80 to 85% cases, 80 to 85% cases of Japanese encephalitis are in children under 15 years, okay? It doesn't affect infants. That's why you saw even we started the first dose of Japanese encephalitis at 9 months. Clear? So, this is your catch-up immunization. Now, second scenario. If there is a 2-month-old infant and it has missed OPV 0 and BCG, what now? So, you can still give BCG if under 1 year. OPV 0 not needed now, start schedule from 6 weeks. Okay. So, OPV 0 can only be given till 15 days. After that, if the child has passed that window, you can also start at 6, 10 and 14 week. The first, second and third dose. Okay. And BCG you can give till 1 year of age. Okay. Now, this was everything about your vaccines. Now, before I end this lecture, let's talk about how to approach a short note if it comes. So, if it comes on types of vaccine, right? If be it a live vaccine or your newer vaccine technology like your mRNA, edible vaccine, these mRNA edible vaccines mostly MDs get in their exam, right? How do you approach this? If it's a, it's a high marks question. Always start with the definition of your vaccine. That is, vaccines are immunobiological preparation. Okay. Then you come to the types of vaccine. In types of vaccine, always make it in a chart. You make it like first, live attenuated with example, killed with example, subunit. In subunit also 1, 2, 3, the polysaccharide, the conjugate and the protein. Then toxoid vaccine and newer vaccine technology. Example, one example of each. Then you start writing about your, either it's live vaccine or mRNA. Always write in bullet points. Don't write in paragraph. Or if you get like a passive immunization, okay, start writing everything in bullet points, and at the end, give a difference, give a table with at least 3 to 4 points of active versus passive immunization with example. In passive also, you have to also, add the point where and which condition we are using passive immunization. Okay, that's all. Thank you.